Hello everyone. Welcome back to Touch Tank Tuesday. Hopefully you can hear me okay. My name is Olivia and I am the education lead here at the Deep Bay Marine Field Station. We are a research and education facility that's part of Vancouver Island University. And a lot of our research focuses on shellfish aquaculture, really the whole shellfish aquaculture industry. And really our local marine environment. So there's a lot of research that's going on here. But today we will be looking into our touch tanks to talk about another crazy critter in there. And hopefully you can see it here. You probably don't know what it is, but that's okay. I will give you some hints. So today we are going to dive into the world of microscopy. So we're going to be looking into the microscope to see something at very, very high magnification. So scientists really do rely on microscopes to be able to see and study things that we couldn't see just with our eyes. And so today we're going to look at sediment, so the sand, and what kind of things we can find in the sand of our touch tanks. So this is some sand here, just in a petri dish. So this is some fine-grained sand. And we have 3 touch tanks. One of them has a sandy bottom, and 2 of them are either rocky or have lots of oyster shells on them for the critters that are in them. In our sandy bottom touch tank, we have anemones, we have some sea cucumbers, and a bunch of other creatures. Ooh, sand dollars, because sand dollars love to burrow. We also have a bunch of clams, but you actually have to dig into the touch tank to find them. But today we are going to be talking about Foraminifera. So this, what you can see here, is you're looking into a microscope, and we have our document camera that's projecting it up for us. So you're looking into the microscope at this very, very tiny creature, and this is a Foraminifera. But Foraminifera is a group, and it's a very long word, so I'm going to just call them forams for now. So I have been collecting some sand from around Deep Bay, and inside the sand I was able to find this beautiful creature, and there are actually a couple different species that I will show you today. This beautiful tiny creature, and this is a foram, and forams are hello. So forams are really amazing creatures that we would not be able to see without a microscope. They can range from being very, very, very tiny to actually being almost 20 centimeters in size. But they're single-celled organisms. So they have this one cell, and from that cell, they create a shell. And so you can see the shell here, and we call it a test. And if you remember from some of our previous sessions, that word should be familiar. A test, just like our sea urchins. So here is the test of a sea urchin here. So that is very similar to what we're looking at on the microscope. So the sea urchin test is made out of calcium carbonate, and so is the test of a foram. And but where sea urchins are made of many cells because they are large eukaryotic organisms, they're multicellular, this Foraminifera is made of just one cell. And what it does is it takes that calcium carbonate out of the environment and it creates its own home. So that's what this test is. As we go along, I will basically talk about Foraminifera, where they live, why they're important, and why scientists find them extremely interesting and good for monitoring our marine systems. If you have any questions, please do post in the comments section and I will try to answer them live. If I can't answer them live, I will come back later and hopefully be able to answer them for you. I particularly love Foraminifera. I did some research on them previously and was very excited to find them here in Deep Bay. Okay, so let's begin. Foraminifera is a phylum. So it's a subphylum. So basically anything that's in this group, Foraminifera, is single cellular. They create a test, like what you see here, so that shell. And that shell is multi-chambered, so it has many little chambers. And hopefully you'll be able to see it if I move it around a bit. But just like a seashell, Foraminifera can be in a spiral shape. They can also be elongated, so they could be long and look like a long shell. But they have all of these different chambers. And basically what happens is that it's this one cell, but the cell goes around the shell. So if this were the shell, the membrane of the shell, or the membrane of the species cell goes around the shell, and the shell is on the inside. And then on the inside of the shell, there are all of the organelles. So all the organelles that help make it function properly. So all of those are on the inside in those chambers inside the Foraminifera. Foraminifera as a group has 50,000 different species. So there are so many, but 40,000 of those are extinct. So we only have fossils of them. The other 10,000 are still alive, and you can find them all around the world. Foraminifera can live on the seafloor, so they will be stuck to the sand, they would live in the sand, they would stick to rocks, or they can even be something called epiphytic. Epiphytic? I think I got the word right there. And that means that they are on plants. Oh, hi James, welcome. So they actually will stick to plants and live on plants. And these are called benthic foraminifera. So that's that group. And most foraminifera are called benthic foraminifera. And then we have another group, planktonic foraminifera, and those are a little bit bigger, and they kind of look, a lot of the species look like little popcorn. And they float in the water column, and they spend their lives floating about. So those are those two groups, but most foraminifera actually live in the sand. So if you go out and you're looking in the sand, you might need a microscope to see them. But if you looked closely enough, like I did here, you could find foraminifera there. And now the one that you're looking at is actually not alive because it's just the shell. So this foraminifera is not alive. You're just looking at the shell that's there. Foraminifera live in marine environments. Some of them live in brackish water. So if you remember, brackish water is that water that is kind of a mix between salt water and fresh water. It mixes together. You can find brackish water in like estuaries, in areas with a lot of freshwater input. Our water here in Deep Bay kind of borders on brackish. It's just a little bit more fresh than supermarine water would be, like saline water. And so some species do live in that environment, and other species, I think there's a couple that actually live in freshwater. So they can be quite spread out. Ooh, so James has asked, how do they breed? So they breed in two ways. They breed sexually and asexually. So they have very complicated life cycles. A foraminifera will go through, they alternate generations. So one generation would breed sexually, the other generation then would breed asexually, and then it would continue to alternate. So they have this alternation of generations. It is kind of complicated how they breed, so we won't get into it too specifically, but that's generally how they would breed. And there are many of them, and they're all over the world. So they're in all the different oceans, and which is great for scientists because that means that they can study it all around the world. And so foraminifera are quite interesting in the fact that they have these body chambers. And if you look closely, you might not be able to see. I'll see if I can flip this foraminifera over for you. Let's see. I might just end up hitting it. Okay, so here's our forum. And here we are. Oops. So this forum is actually less than 63 microns in size. So less than 63 micrometers, if you could think about that. And there we go, it's flipped upside down. So this is the backside of the foraminifera. And as you can see, it kind of has this center. So it spirals around a center. That center is where the chambers kind of go out from. the chambers go around. And if you were able to see in a bit higher quality, you would be able to see all these tiny little perforations, so these tiny little holes in the test. And these tiny holes, when the Foraminifera is alive, these tiny little holes have long projections that come out called pseudopods. And these pseudopods are basically hundreds of tiny little feet that come out. And that's just like the sea urchin, if you remember, and it's just like the sea stars where they have tube feet that come out and help them move. The Foraminifera actually has these pseudopods that go out and they help them to move. So they'll actually crawl along the sea floor. They'll use them for feeding, so they'll be grabbing things to eat. And they'll use them to hold on to things. So if they want to hold on to those plants, if they want to hold on to rocks, they can do that. How do they find a mate, James asks. I'm not sure how they find the mate, but they are found in really high amounts. So there would be a fair number in one area. And so if they did mate, and we're talking about Benthic Foraminifera, so the ones that live in the sand, they move in the sand, they would be living in communities and colonies. So they would have a pretty high population in that area if they did live there. So I assume that they would mate within that population. And then if they ended up moving elsewhere, if there was another population there, they would mate with them as well. And so for eating, Foraminifera, they're so tiny. They're very, very small. So all of these ones are less than 63 micrometers. That's why we need the microscope for them. But they actually can eat algae. So some species will eat just diatoms. And if you've seen our conversation on algae, they'll just eat diatoms. Other ones might eat detritus. So if there's any organic matter on the bottom of the seafloor that's breaking down, they'll eat that. Some are big enough to actually be able to eat copepods, which are those very, very tiny crustaceans that are in the water. So they really have a huge diversity in what they eat. And then when we talk about what eats them, they have many predators. So lots of fish actually eat Foraminifera. So these fish would eat these Foraminifera accidentally, and scientists believe that they actually eat them as a source of dairy, a source of calcium. So because these tests are made of calcium carbonate, kind of like this one, that's how fish might get their calcium, by eating Foraminifera. So they're kind of a dairy product of the ocean for these fish. And so that's one thing that would eat Foraminifera, but there are also snails that eat them. So there are snails that actually specialize on eating Foraminifera. And scientists have actually found that some species will prefer different species of Foraminifera. So they'll eat solely on one species or another. James asks, where do they live in the ocean? So they actually live pretty well everywhere in all the oceans. There are two groups. One group are Benthic Foraminifera. Those ones live in the sand. So live ones are usually found in the first 10 centimeters of sediment as you're going down. And they will actually move with those pseudopods up and down in the sediment to different layers of sediment throughout their lives, and actually seasonally. So they'll live in the sediment. Then the other group, which are planktonic, so the planktonic Foraminifera, there are much less of those ones, but the ones that do live in the ocean end up floating in the water column, and that's probably where fish might eat them, and that's where they would eat probably algae that's in the water column there by using their pseudopods. Some Foraminifera are actually also filter feeders. So like we talked about with some of the other species that are filter feeders, kind of like sea cucumbers, those ones are filtering the water, like oysters that filter. Foraminifera also might filter water depending on the species, and they would eat from that filtered water. So those are the predators, and when Foraminifera die, they'll look just like this. So this is a test. It's just their shell with nothing on the inside. So the shell is left in the ocean, on the ocean floor. And what happens is over time, as those shells stay on the ocean floor, the sand will cover them and more sand. And throughout time, we'll have layers and layers and layers of different foraminifera species, depending on what was alive and died at certain times, which is why they are so important for something called biostratigraphy. So we'll talk about stratigraphy. Stratigraphy is a field of studies, so a field of science, where scientists look at how the environment has changed over time. So, you know, if we look out and we see where our ocean is, well, thousands of years ago, that shoreline might have been in a different place. And the way scientists are able to study that is by looking at the different layers of sediment and layers of rock that are found all around, even on land, because maybe there was an ocean environment where we see land now. Maybe there was an ocean environment millions of years ago. So it's really quite cool. So they look at all of those layers. And then biostratigraphy is where they look at the different fossils that are found in each of those layers. And by looking at the fossils, they're able to date it. So when was this part of the rock this way? So how many years ago did it look this way? And they're also able to understand what the environment was like. So if one species of Foraminifera might like very saline, so very, very saltwater environment, if they are found in an area, then scientists would know in that at that time where that Foraminifera lived, it was very, very salty. So they're able to basically draw lines between where these species live now, where they used to live, and they're able to connect what the environment looked like many, many years ago. So it's really cool, and it's really complicated what they do, but Foraminifera are very, very special for doing that. And they're very special because of how many there are. So you'll find lots of Foraminifera in these samples, and you'll find them all over the world. So you can compare them all the way around the world. Does anyone have any questions so far? I'm going to make this big, and I'll show you some of the other species I found here. So this is just one species, but I do have a fair number of them here. Let's see. So this one over here looks like a different species. So this one is a lot clearer. It has a bit of a different shape. Oh, and then there's this one here. Let's see if I can zoom in. Yeah, so you can see all those chambers. This one kind of looks like a bit of a flower. And you can see all those chambers going around it. And what I will also do is kind of show you what my setup looks like here. So this is a microscope. This microscope is a dissection microscope. That means that it's not a compound, so it's not looking at a slide up here if you've seen them previously. So this is a dissection microscope. We have this slide here. And so if you look, this is what those Foraminifera look like from your regular eye, and that's what they look like under the microscope. So they look like just tiny little grains of sand. And I'm connected here with the document camera. This document camera is connected to the microscope, and we're able to look into it there. So really is necessary to have. Okay, so James has asked, how many species are there? So there are 50,000 species of Foraminifera. So there are lots, but 40,000 of those are just in the fossil record. So they went extinct many years ago. 10,000 Foraminifera species are still alive, and they can be found all around the world. So these ones here, I think I have maybe 3 different species, and I just don't remember which ones they were. It takes a lot of time. What you have to do is look at to be able to identify which species it is, you have to look at the chambers. So how many chambers they are, like the shape of the chamber. So if it goes in a circular spiral pattern, if it is long and spiral, how many chambers it has, where its aperture is, so the hole that opens up into them. So there's an aperture. You need to know where that is, whether the spirals go up, whether they stay in the same line. So there's all different body features that you would look at. You would also look at what the test is made of. So this test is made of calcium carbonate, but it's a calcium carbonate that makes it very porcelain. It looks like porcelain, it's shiny. There are foraminifera that actually have this test made out of chitin, or all of these tiny little sand grains. So they really look like sand grains, which is amazing. So they have different types of tests made of different things, and they're shaped in different ways. Hi, welcome. Does anyone have any other question about the foraminifera? I know it's not a very fast-moving item today. Let's see if I can find another one here. And so these are all picked out of a sample of sand. Okay, so this one is upside down. And I know it's a bit difficult to see. This one is upside down. But you can see actually at the center of the foraminifera, that is where all of those little tiny pores are, and that's where those pseudopods, so those tiny legs, come out. Okay. Let's see. Jim has asked, please spell. So foraminifera. Hopefully you can see that. I have commented back saying foraminifera. Foraminifera. So it is quite a long name, but basically it is very amazing what you can find in the sediment out in the ocean. So when we're walking around and just looking at these sand grains, there's actually an entire community, an entire world living in the sand that we just can't see. And they're really important for processing. So that detritus I was talking about. So when you have matter that floats to the bottom of the floor and it's decomposing, these will help with that. The foraminifera will help with breaking down that detritus. So they really, really are an important part of our entire system. Was the clear one earlier live? Is it hard to find a live one? So all of these ones are dead. What scientists do usually is they would collect a sample of sand. They would kill everything that's in it to preserve it. And what they would do would be to put a dye in. So they would actually stain the whole container. And that stain, it's called rose bangle. That stain is actually used to identify living tissue versus dead tissue. And so the stain will turn pink anything that is alive when it was collected, and it would just leave blank anything that wasn't. So usually when you're doing a study on these critters, the only way you'd be able to tell whether they were alive or dead, unless you're looking at them in the water where you could see those tiny legs coming out, the only way you could identify them would be by staining that tissue just to see if it was alive or not, because they are so small. They're quite amazing. Yes, Jim. The ones that are on here, they're all dead. And I'm not sure if they are alive when I collected them, just because I wasn't able to look at them under the water. I think you'd probably have to dye the water as well to see them, or have a really high magnification microscope to see them moving about, just because you can see they're white. So having a dark background is really necessary. But then you would find so many other things in the sample that it would be really hard to really find one foraminifera. Hi, you're welcome. And so scientists will use them to study the environment. Basically, each species has a particular set of conditions that it loves. So let's say one species really likes areas that have high currents. The water moves, and so they're found there. And we understand that that's where that species is. If you took sediment samples from different areas, you could know what area had really high currents because that species was found there. And so that's why scientists kind of create this baseline understanding of where these foraminifera live so that they can study the past. Studying the past climate and the past ocean is called paleoceanography. So that's kind of studying how the ocean has changed over time since the beginning. So what scientists would do would be to take sediment cores. And this kind of thing happens with drilling programs. So lots of companies that are looking to find oil, really ocean drilling, they would drill down and create these cores. And these cores have basically layers of sediment, like I talked about before. And each of these layers would represent a different time. And to know what time it was that that layer was created, you could use the tests of foraminifera because they're made of calcium carbonate, and you can use isotopes. So if you tested the test of a foraminifera, you'd be able to carbon date it basically. So when was this created? And that way you would know what time that layer of sediment occurred and what time it was laid down on the ocean floor. So it really is complicated and really a wide field of studies that are happening right now with foraminifera. Not a huge number are happening in Canada. Many of them are happening in Europe. There are a couple European institutes and universities, but they are quite an interesting creature to look at. And it's really just amazing seeing it and knowing what's in the sand. It's not the only critter that's in the sand, but they are pretty cool. I will try and put onto Facebook a couple high quality photos. I know they're kind of hard to see right here. It was interesting trying trying the connector to the document camera, but it is kind of hard to see on the screen. So I'll try and take some photos with our really high-end microscope so that you can see a little bit better. Okay, well, if there aren't any more questions, thank you so much for joining us today. I hope you learned a little bit about foraminifera, this mysterious creature that's in the sand. Oh, Jim's asking, are you doing any sample cores there at Deep Bay? No, so we are not doing any. Thank you. We are not doing any sample cores here at Deep Bay. I personally am walking around and collecting small sediment samples so that I can see what species we have here. And that's just out of my own curiosity. We are not currently studying them, but it is cool to know that they are there. Thank you so much, everyone. Thank you for joining us. We will not have a session next week because we have a class coming in to look at sea star wasting syndrome. So that might be a topic for the week after, but I will see you in 2 weeks then. Bye.